Okay, so we are modeling here meiosis, and this is the very beginning of prophase 1, when the chromosomes start to duplicate, creating the sister chromatids that are held together by cohesin, which creates sister chromatid cohesion, and it's gorgeous. And then also during prophase, the homologous chromosomes start to line up next to each other, and they line up by length, and this creates tetrads. And then something called crossing. Oh, no, actually, this also the independent assortment creates genetic variants, because they could also be like that, and that would create these all create different cells, which is like a whole big thing.
And then while they're here, they start to cross over, where they exchange genetic material, and then when they cross over, it's called the chiasma. So as they exchange genetic material, this is also mixing up the genetic variants.
All right, let's go. Okay.
The cells don't find this nearly as difficult as we do. Oh no.
And then after they cross over, the chromosomes line up in metaphase and then start to pull apart in anaphase 1. And the microtubules pull them to the sides of the cell. And then in telophase, the two cells start to separate, and cytokinesis separates them. Yes.
And so then we have the beginning of, meiosis 2.
And then the chromosomes
we'll talk about
again,
1.
during metaphase 2, they start to line up again in the cell. They do that in both cells. And then the cohesin will separate, and the microtubules will again pull apart the sister chromatids into two separate cells. They'll do that on both sides. And then after telophase and cytokinesis, they'll make their own separate cell, and then we'll end up with 4 haploid cells.
All made up of non-sister chromatids.
And right here are the centromeres, and